Hey, hi everyone. So we'll be starting shortly. We just had a long interaction session with Professor Partha Majumdar and he'll be back shortly for the session. So all of you have access to the Q&A, right? So you can ask questions as the presentation goes on, and you can. So Professor Partha can answer them while he is speaking, or at the end, and so on. So feel free to ask questions. So we have Professor Partha here. Hey, hi.
Yeah, are you able to hear me? Is my audio good?
Yeah, very clear. Yeah.
All right.
So you are able to see the Q&A panel, right? So the participants may ask questions there, and you can choose to answer them either during your talk or at the end, however you deem fit.
I would prefer at the end, but where is the Q&A panel? Participants, chat, and Q&A. Okay. All right. It's empty. Okay.
And hey, Himanshu, hi.
Muted. Hi, Partha. This is Himanshu here.
Hi. How are you?
I'm good. How are you?
I'm good. Good to see you.
Yeah, actually, let me switch on my video again. Hi. Hi.
Good to see you.
So you had a good session with the students, I believe, right? You had a
I
session.
guess so. I learned more than I actually delivered.
Okay.
Yes. Some very interesting work going on there. So that's very nice.
Yeah. Let's see.
Good.
So Karthik, are we waiting for students to join? How many have joined? 32 already. Should we start?
Yeah, we can just
You
wait.
have another meeting, Professor Partha, right?
Yes. Today is a very busy day. Started at 9:30 and we go on. Yeah. So I guess many of you know that we are having this Frontiers of Science Symposium of the
Yes.
Academy, and we're doing the single cell or human cell atlas, which is essentially single cell genomics. And we have a series of talks. We had one this morning, followed by the secretary who spoke, and then we have two more technical
In the
talks.
evening. Yeah.
Yes.
Okay. So
No, I'm ready. So whenever you're ready.
Karthik, you want to start the?
Yes, I'm just bringing in a bunch of participants as panelists, so they can ask some questions at the end maybe and so on. But
Okay. But that you can keep on doing as the seminar progresses.
Let me have the privilege of introducing Partha. So as I think Partha is one of those people who require no introduction, but I'm pleased to introduce him. He is the National Science Chair and President of the Indian Academy of Science, and a distinguished professor at the National Institute of Biomedical Genomics at Kalyani. And he's the founder of the NIBMG and concurrently a professor at the Indian Statistical Institute. And he's done a lot of contributions to human and population genetics, from the structure and evolution of populations to genomics of diseases, and of course the development of a number of statistical methods for genomic analyses. He is a Sir J.C. Bose National Fellow, and he's an elected fellow of all three science academies in India, a rare honor, and of the World Academy of Sciences and the International Statistical Institute. So he is more of a statistician by training, but as he was telling our students just a while ago, he stopped answering others' questions at some point of time and started asking his own questions, and building a very strong and independent research group over the last several decades. So he's also a council member of the Human Genome Human Genome Organization, and most importantly, the Indian National Coordinator of the ICGC, the International Cancer Genome Consortium. He's also served on several boards, including the International Bioethics Committee of the UNESCO, and has many, of course, publications to his credit, and is a recipient of several awards and medals, including the TWAS Biology Prize, the G.D. Birla Award for Scientific Research, a Ranbaxy Research Award, and the New Millennium Science Medal. So, and above all of this, if you've had a chance to interact with Partha, you will realize, how absolutely down to earth and cool he is. And I'm sure The students will attest to that, having just had a less than 1 hour conversation with him a few minutes ago. And it's my pleasure to invite him to the 6th IBSE colloquium. And he's of course going to talk to you about what everybody wants to hear about today, the the virulence of COVID-19. So over to you, Partha.
Thank you, Karthik. Thank you for those very kind words. As a matter of fact, I should start with an apology to Karthik. Professor Karthik Raman has been asking me to give this talk for quite some time, many months now, and a little bit of forgetfulness also because of other kinds of activities that I do. I've been postponing it and telling myself, yeah, I have to reply to Karthik, and the last mail was something that really put me to shame. So I said, no, I have to do it. And believe it or not, last night I realized that I had announced this Frontiers of Science Symposium, and something needed to be done because I was supposed to be engaged with all of you from 3 to 5:15. And I couldn't do that because at 4:30 I have to start the other symposium. So fortunately, everything worked out. So we just moved it up by an hour. And this talk remains at exactly the same time that, so it's a little bit of forgetfulness. I don't, use a secretary. I'm my own secretary. So, and the kind of work that I do, the various disparate kinds of work that I do makes me forget to a little extent. But be that as it may, I'm absolutely glad to be here. The first thing that I will say to you is that I have no slides. I'm just going to narrate a story to you. This is based on, I'm going to make some very, very general remarks. I'm told that the audience is not very homogeneous, so there's some amount of heterogeneity in the audience. So I'm going to talk some general characteristics and then home into our own work in this particular area where we made an observation. That observation took us to doing a little bit more work, but it's all the latter part of the work is more biological. But nevertheless, I think I will be able to narrate a somewhat complete story about one aspect of this pandemic. So I'll start at the beginning, and I'm pretty sure that all of you know that SARS-CoV-2 is a virus, and I'm sure that you also know that viruses are obligate parasites in some way. They cannot live by themselves. They have to be able to get into somebody else's cell and hijack that cellular machinery to their own advantage, essentially to procreate. Otherwise, they are dead. So it is to the advantage of the virus. It is a necessity for the virus to get into somebody else's cell, the host's cell, and then use that machinery to their advantage. So they are, such as the SARS-CoV-2 being a virus is an obligate parasite in some ways. They are, we can't really characterize them as a living organism, primarily because they are unable to procreate or reproduce themselves by themselves unless some other cellular machinery is available to them. The SARS-CoV-2 is the largest of all viruses that we know. It is an RNA virus. The length of its RNA sequence is 30,000 nucleotides, which makes it a very large RNA virus. The viruses are usually these nucleic acid sequences wrapped in a protein coat. This particular virus is wrapped in lipids, and that's the reason why we are asked to wash our hands every so often. Soap will melt away the lipid layer, disintegrate this lipid layer, expose the naked RNA, and an exposed naked RNA can't live for a long time. They get cut into small pieces, and there goes the virus. That's the natural death of the virus through washing our hands or rubbing alcohol on our hands. That's essentially what it does. It melts away the lipid layer. This particular virus is also very interesting because it's a positively stranded virus, and the positively stranded virus really means that it can skip one step of its reproduction process and it's ready for replication. So it's like an mRNA that's already poised to replication, and that's what this virus is all about. So it's a positively stranded RNA virus, quite long and has multiple genes. In particular, it has, you have seen the structure of the virus. That's why it's called a coronavirus. It is like a round ball with lots of spikes, those spike proteins, and I'll come to those spike proteins in a minute or in a few minutes. And those spike proteins are absolutely critical to the spread of the virus. What I'm going to talk to you about is the evolution and spread of the virus. So let me first talk to you about the first part of my talk, which is about the evolution of the virus. I don't have too much to say on the evolution of the virus. The way that we study evolution, or at least those of us who believe in molecular evolution, the way that we study evolution is to sequence nucleotides or sequence the genome of any organism for that matter. And we sequence a large number of organisms and then essentially compare the nucleotide sequences of the genomes. And we say that those two genomes that are very close to each other are similar, and those that are close, meaning identical to each other, are identical nucleotide by nucleotide are similar. And depending on where the two different viruses come from, we can even sometimes fix which is ancestral and which is descendant. So in this particular case, if we, again, just to begin my story, as we all know, the SARS-CoV-2 was the first infection was in this lady who was working in the seafood market in Wuhan. And she came down with, the most characteristic features of this disease, of this coronavirus disease, which is coughing and fever and so on and so forth. So she came down with all of this, and they took a throat swab, and they were able to find out that her lungs were infected, and they isolated this virus, and they sequenced the virus, and it turned out to be a coronavirus. Incidentally, this lady recovered, and she's up and kicking still in Wuhan. So this coronavirus was sequenced simultaneously, not simultaneously, even before then, there were some coronavirus sequences that were already available. These sequences, a few of them were from humans, but the vast majority of them were from other animals, especially the bat. The bat is home to a large number of viruses. We don't really know why the bats are home to so many different kinds of viruses. I'm told that the count is 67. The bats are a home to 67 different kinds of viruses, including some species of coronaviruses. So what was, and those sequences were already available. And so when the first sequence of the SARS-CoV-2 was made, was obtained from this isolate that was isolated from the viral isolate that was obtained from this lady who was working in the seafood market. This sequence was compared with a whole host of other viruses and virus-like particles and coronaviruses, a few that were already available. And it turned out that this SARS-CoV-2, if we looked at compared nucleotide by nucleotide, the identity of nucleotides of SARS-CoV-2 was very close, was the maximum with another coronavirus that was in the bat. And since this coronavirus was reported from the human only recently, in December of last year, it was only logical to conclude that the bat coronavirus is ancestral to the human coronavirus. It was 97% identical to the human coronavirus SARS-CoV-2. This is not the first coronavirus that has infected humans. There have been other coronavirus infections. As a matter of fact, even though there is a bias of ascertainment, we know that there are 7 different coronaviruses that have been isolated from the humans at different time points. 4 of these viruses actually don't cause major symptoms. They cause just flu-like symptoms. You sneeze for a few days, and then it essentially it retreats and it gets resolved by itself. There are 3 that we know, including the SARS-CoV-2. There have been 2 other major outbreaks that cause major disease and fatality. This is SARS-CoV, again, something that an outbreak that took place in Wuhan in 2003. But that was an outbreak. It didn't even expand into an epidemic, and certainly didn't expand into a pandemic. So this was an outbreak that sort of was contained by itself. It was called SARS-CoV. Ideally, it should have been called SARS-CoV-1, At that time, nobody imagined that there's going to be a new avatar of that virus that we would call as SARS-CoV-2. So anyway, that's SARS-CoV and this is SARS-CoV-2. In the interim, we had another coronavirus outbreak, which was in Saudi Arabia, in West Asia, and that's called the MERS coronavirus, Middle East Respiratory Syndrome coronavirus. So there are these three coronaviruses: SARS-CoV, MERS, and SARS-CoV-2. These are the three coronaviruses that have created a lot of problems for the human. I will come back to a few more characteristics comparing these three in a few minutes, but in terms of the evolution, like I said, that by comparing the identity of the nucleotide sequences, we reason that the bad coronavirus must be the predecessor, ancestor of the human coronavirus. It was accidentally something very interesting was found by a few people. They found about 200 kilometers away from Wuhan in the morning near a bush. They found two anteaters. These are pangolins, scaly anteaters, two anteaters that were dead. They were accidentally detected, and these two anteaters, when they were discovered, dead anteaters, when they were discovered, it was found that they had a lot of froth in their mouth. So they had a lot of froth coming out of their mouth, and so it was evident to anybody who knows, what this froth meant, that their lungs were infected, and therefore an autopsy of these pangolins was done. They isolated appropriate fluids from the lung, cultured them, and not cultured them, whatever they did to that. I suppose they isolated whatever. And sequenced, they found coronaviruses there and they sequenced those. And it was, again, by comparison of sequences, although the jury is out, we are still not 100% sure through this comparison of sequences. It was reasoned that the coronavirus from the bats didn't come to the humans directly. It came to the pangolin, so there was a zoonotic crossover, a species crossover from the bat to the pangolin, and then from the pangolin to the human. Now, how did it come from the pangolin to the humans? We probably are closer to the bats because oftentimes, there are these bats in trees in our neighborhood and so on. So we are probably closer to the bats in closer proximity to the bats than to a pangolin. Turns out that in China, in this same seafood market in Wuhan, they also sell exotic meat. And one of the kinds of exotic meat that they sell are meat of pangolins. And so there are these workers, seafood market workers, who actually butcher pangolins and scale them and sell the meat. And that's an exotic meat, apparently sold at high prices. So we normally don't come in close proximity to the pangolin, but once we do, and if the pangolin is infected, then it can through certain kinds of mutations that it might accidentally acquire and change over, may have a zoonotic crossover into the humans. So, like I said, the jury is out about the belief in most microbiologists and people who carry out viral DNA sequencing and comparisons believe that it was bat to pangolin to human. So we'll just take that model for, so there is usually an intermediate host before it crosses over to the humans. And in this case, the intermediate host is most likely pangolin. Now, it's not just the sequence comparison, but there are also other reasons why many people believe that it must have been the pangolin. The pangolin must have been the intermediate host. All right, so this is what I wanted to say about the evolution of the virus. I do want to spend some time about the spread of the virus. And the first thing that I would like to state is that, and I've written this, I've written a number of popular articles on SARS-CoV-2, both in terms of, several aspects of COVID. But anyway, in one of the articles that I've written, I've called this virus, SARS-CoV-2, as a very kind virus. It's almost non-pathogenic. It's not almost non-pathogenic in the sense that it does infect us. It does create some problems in us, some major problems in some, where you have respiratory distress, you have fever and all kinds of stuff. But anyway, it doesn't kill very much. So I want to first give you a comparative figure of how coronaviruses kill humans. And the reason why I've called this virus as a very kind virus. If you compare, like I said, that there are 7 different coronaviruses that we know that infect the human. 4 of them are kind of benign. They don't create any major problems in humans. 3 of these, the SARS-CoV of 2003, MERS of 2013, and SARS-CoV-2 of 2019. These are viruses that create a lot of symptoms in humans and sometimes kill. We know that SARS-CoV-2 is killing on an average 1% of people who are infected with SARS-CoV-2 virus. And of course, there is some age difference. Elderly people, the killing rate in the elderly people is more than that in younger people, and so on. But on an average, it's across all age groups, it's killing about 1% of people that it infects. If you look at the SARS coronavirus and the MERS coronavirus, both of those were much more deadly, much more unkind compared to the SARS-CoV-2. SARS coronavirus 1, if I may call it 1, SARS coronavirus 1 on an average killed 11% of the people that it infected. The MERS coronavirus killed 34% of the people it had infected. And remember, this coronavirus is only killing 1% or less of the people that it infects. So in some ways, it is a very, very kind virus. On the other hand, this is the first pandemic of a virus that we are, of any infectious pathogen, that any of us, either in the audience or listening to me in the audience, or living on this planet, has ever faced, because the last pandemic was in 1918, and nobody who was born in 1918 is probably alive today. There may be a few people who are alive, but very, very few people are alive. Very, very few people have seen the last pandemic, which was also a flu. This is also, like, it's very related to the influenza symptoms and so on. So it's the last pandemic was one of Spanish flu that was in 1918. So we have never seen a pandemic. So on the one hand, this particular virus is a kind virus in the sense that it doesn't kill many people, but on the other hand, it infects many people. So it's a master spreader, but a poor killer compared to the other two. Viruses, you will imagine that really it's not to their advantage to kill their hosts. They need the host to be able to procreate. They need the host to be able to procreate and then spread. And the more they spread, the better they are in terms of a Darwinian selective advantage. So why would they want to kill the host? So it is, to its advantage that it will not kill the host. And this particular coronavirus is an epitome of that. Any pandemic, any pathogen or any organism that creates a pandemic will have this characteristic property. It's not going to kill too many people. Although many more people died of the Spanish flu because it was both highly virulent as also a master spreader. This is not a virulent organism, but a master spreader. So it spreads very well. I want to spend a few minutes trying to discuss. We are talking about spread. At some point of time, we need to know, very often, I'm sure that all of you who are in the universities, etc., the common man would have asked you, when are we going to get rid of this? When are we going to, come out of this pandemic? So the answer is not easy because, the answer is not easy, period. To be able to answer such a question, we need a statistic, we need a quantitative measure by which we can track whether the spread of the pandemic is coming down or whether it's still growing. One of the key statistics that you may already have heard about is called R0. So I want to spend 2 minutes explaining what R0 is all about. And I know that many of you already know the definition. Many of you know how to calculate this, etc. But the R0, again, R0 is not a very simple statistic. It's complex because it's a product of 3 other statistics, 3 other quantities. Two of them we can't control, one of them we can control. And I'm not going to get into the gory details of the three components. Be that as it may, we can only control one component. So what does R naught represent? R naught represents, R naught is the value of R naught represents how many uninfected individuals can an infected individual infect. So if R naught is like, takes the value 5, that means on an average, one infected person is infecting 5 uninfected persons, which means that the virus is spreading very rapidly. If the value of R naught comes down to about 3, then you know that it is the spread is coming down. R naught value of 3 means one infected person is able to infect 3 uninfected people. When R naught becomes equal to 1, so it's 1 infected infecting 1 uninfected, which means that we have reached a plateau. When R naught comes below 1, then we know that an infected person is not even able to find one uninfected person to infect, and therefore the pandemic is going to slow down and is going to die a natural death. So what determines whether an infected person can find uninfected people? When you're infected, you grow certain kinds of immunological proteins in the body, and they sustain for some time. Some of these have memory, such that when the same virus again attacks you, you don't come down with these with the infection. You don't allow that infection to happen, and therefore you do not have you don't have any symptoms. You don't suffer from it. You don't get reinfected. Of course, the reinfection does not last for a lifetime for all kinds of viruses. In this particular for SARS coronavirus 2, we don't even know how long this memory lasts or how long the immune system remains active in order to ward off reinfection. We don't know that yet. We don't have a firm estimate or a robust estimate yet. But be that as it may, for some time it will not be one will not be infected. So this the reason why, the way that we can bring down the value of R naught, that is something that is controllable in our hands, is if we have a large number of people in the population who are already immune, who have already gained immunity against SARS coronavirus 2. One way to gain immunity is natural, which is that this coronavirus will infect a large number of people. Those individuals will gain immunity and therefore they cannot be reinfected. And so one is natural ways of gaining infection. The second is an artificial way of promoting infection, promoting immunological restraint or to infection, which is through a vaccine. We still don't have a vaccine for SARS coronavirus 2. As all of us know, feverish work is going on at a great pace, but we still don't have a vaccine. And people who are vaccinologists don't believe that the vaccine will come within this year at all. So we will have to live with this virus. The virus will spread naturally until such time that R naught becomes less than 1, and we have no choice. We can impose lockdown, we can do whatever. We can slow down the spread, or we can stretch the spread for a longer period of time, but that's all we can do. We can't, until we gain immunological ability to ward off the virus and gain that memory to be not be infected, this is going to happen. So we might, through lockdowns, we might stretch the period, but that's all we can do. We can't do anything more. We can stretch the period and hope that during this period, a vaccine will emerge. Those of us who have not gotten the infection will get vaccinated and will generate artificial immunity to the virus. All that is in the wish list. But the fact is that if the vaccine doesn't come through the natural infection process, many of us will get infected. But this is a kind virus, so it is not going to kill many people. We will all recover. I exclude myself because I'm probably the oldest of all of us assembled here. So I will probably not recover if I get infected. So I have to be a little bit careful and not expose myself so much. But there's no guarantee that I will not be exposed and not be infected. So R naught is a statistic that we track in order to find out whether the epidemic or the pandemic is coming under control. and it will come under control when R naught is less than 1. When R naught is, when the proportion of people in the population becomes, when about 2/3 of the people in the population become immune to the infection, then R naught will become less than 1, and this can be proved. This is not a take it from me statement. It can be algebraically proved that you need about 67% of the population to be infected with the virus for R naught to get below 1. So that that is called herd immunity. When the community, in the community, the virus has spread and many people gain immunity in the community, then that's called herd immunity. All right. So I will come to two pieces of work that we have ourselves done. One has been already published. The other has been submitted was submitted to a journal. It's just come back for revision today, this morning, and we hope that we'll be able to address all of the reviewers' questions and it will be published. So when this happened, when this infection broke out in December of 2019, we got interested, and already for those of us who work in the area of molecular evolution, we jump up and down with joy if we find that there is a repository of sequences of these kinds of viruses. And indeed, we all knew that one of the best repositories of sequences of viruses is the GIS-AID, which has all of the viruses that create flu-like symptoms. So we had access to, and that's a public domain repository. Curated data are passed on to another data set that also has other kinds of computation pipelines called NextStrain. So it was just absolutely a gold mine for us to track this particular coronavirus as it evolved. And we know that the ancestor was in Wuhan, and as the individuals moved out of Wuhan, they carried the virus, and so the virus then spread to other places. But again, the one thing that happens is that as the virus spreads from within the individual and across individuals, evolution of the virus takes place in the sense that the virus accumulates mutations. And so the virus was accumulating mutations. This was obvious by comparing the viral data over time. So as we tracked the virus over time, and the virus was spreading, it spread to Italy, it spread to Europe, other parts of Europe, it spread to United States, it came to East Asia, it came to South Asia, so it spread all over. And we were tracking, and this spread was happening very, very within days and weeks. So when the virus went to different geographical regions and we were doing this phylogenetic analysis, comparing the DNA sequences, looking at the relationships among these DNA sequences, there was also another, normally what we do is these DNA sequences are quite static in the sense that mutation rate is so small. In the human, it's of the order of 10 to the -9 per generation, per nucleotide per generation. In the viruses, it's more like 10 to the -2, 10 to the -3. So viruses evolve much faster. And so as the virus was, as time was progressing, as the virus was spreading to other geographical regions, it was also evolving, and this evolution was fast. So we can't really do a static phylogenetic analysis, which is applicable when the mutation rate is small, but when the mutation rate is high, the phylogenetic analysis from one time period to the next time period will be vastly different. So we need to take this time series differences into account as well, and this is called phylogenetic analysis. So we need to track the dynamic nature of the evolution of the virus, and the dynamic nature is within short periods of time, short bursts of time. So we did this. We were continuously doing the phylodynamic analysis as the virus was spreading to newer geographical areas, and everywhere what we were finding is that there were 5 or 6 different. When we did this phylodynamic analysis, what we found was that after a certain period, within about a month or so, beginning of February perhaps, we already had 11 different subtypes of the virus. So if you do a phylodynamic analysis, and you'll find clusters, and there were 11 separate clusters that were separated at least by 2 mutational events. between clusters, and we were able to identify 11 different subtypes. These subtypes were given names by the GIS-AID. The original ancestral subtype was called O, and they were the derived subtypes, the evolved subtypes were called A, A2, A2A, B1, B2, etc. The names don't matter. There were 11 different subtypes. Now, these subtypes have also been renamed by virologists, but again, nomenclature does not really matter. So while we were tracking, we were finding that in every geographical region, initially after it was populated, seeded with this virus, there are 5 or 6 different types of the virus, subtypes that were circulating. But after about a month or so, in every geographical region, there was one particular subtype that dominated. And this subtype was named as A2A by GIS-AI by virologists. And this subtype, and this domination was something very, very strange. And we noted this, and we asked ourselves that this must be a fantastic Darwinian selective advantage, because it's outcompeting all of the other subtypes and establishing itself in every geographical region. Why so? So we started looking at the nucleotide sequences, comparing it with the ancestral Wuhan type, and finding out what may be the major mutational changes. And what we found was that this particular subtype that was the master spreader, that spread itself so well and outcompeted all of the different, the remaining 10 subtypes, that had an interesting non-synonymous mutation in the spike protein, or near the spike protein. So this is in the 614 amino acid position. It had a change from aspartic acid to glycine. It's called D614G mutation. And this particular virus that was the dominant subtype was D614G mutant, the glycine mutant. So we said that this is very interesting. So what does this glycine mutant do? So now I'm going to combine so this is an observation that's been published, and observation about outcompeting and making itself dominant, and the biology with a little bit more complex biology. And I'm going to combine those two and tell you the last part of my story. So we asked, why is it that this D614G mutant is able to spread itself so well? And can we understand anything about the biology? So in order to understand the biology of a virus, we must understand that, like I said, it has to enter the host cell, it has to enter the cytoplasm of the host cell, hijack the cellular machinery to its own advantage. In trying to enter the cell, there are 2 steps that are involved. One is the virus must go and anchor itself to the cell membrane, to the surface of the host cell, and then somehow it must crack open the cell membrane of the host and enter. So there are these 2 components. There are 2 activities that the virus needs to do. The spike protein helps it go and attach itself to the membrane of the host cell. The membranes of the host cell exhibit different or express different kinds of proteins, and the spike protein must be going and attaching itself to one of the spike proteins that's on the surface of the host cell. And it was found by not us, but even with respect to SARS-CoV and MERS, that the spike protein goes and binds preferentially to a protein that is expressed on the host cell, which is called ACE2, angiotensin converting enzyme 2. Angiotensin converting enzyme 2 is expressed in plenty in the lung, in a particular cell type called type 2 pneumocytes. That's where it is expressed in plenty. So it goes and attaches itself to this particular cell type in the lung, and that's the reason why we get this cough and so on and so forth. That's not the only cell type which expresses ACE2 in large quantities. The enteric epithelium, the gut epithelium, also expresses ACE2 in large quantities, not as much as the lung cells, but it does express in relatively large amounts. And so in about 30% of the people who are infected with SARS-CoV-2, you also find that they suffer from acute diarrhea. And this is probably because the virus has also entered the epithelial cells of the gut. And that's the reason why they have this diarrhea. So anyway, coming back to the lung, so it goes and attaches itself to this particular lung cell, and now it has to crack open the lung cell and enter the lung cell. Now, if you look at the spike protein has two domains. These two domains are called S1 and S2, and these two domains are, when they are joined together, they are essentially inactive. They have to be cleaved, and there's a cleavage site between S1 and S2. It has to be cleaved for the S1 and S2 domains to become functionally active, and these domains then participate in the process of cracking open the host cell wall and enter. And interestingly enough, the cleavage is done by a host protein and not by a viral protein. So it's done by a host serine protease. Proteases are, again, proteins that actually are able to cut other proteins. So the spike protein of the virus is cleaved between the SARS in the cleavage side between S1 and S2 by a protein called TMPRSS1, which is a serine protease expressed by the host, and it goes and cuts that. And once it cuts, then other kinds of things happen. In particular, the virus is able to fuse, the cell membranes are able to fuse, and the virus is able to gain entry into the host cell. So what we looked at was this TMPRSS. As we were doing this phylogenetic analysis, like I told you that the virus spread all over, we also noticed one interesting thing, which is that in Europe or in United States, it established itself as the only type after about a month or so after being seeded in that geographical region. In East Asia and South Asia, that is not to be. In East Asia and South Asia, what we found was that A2A had gained prominence. It had become the dominant subtype, but it didn't outcompete all of the remaining subtypes. There were other subtypes that were also floating around. And so what we asked is, why is it that East Asia is behaving differently from other parts of the world? And we assumed, and this was a hypothesis, that these guys are non-Caucasians. We only have major data from the Caucasians. So maybe the non-Caucasians and the Caucasians behave differently as they encountered this virus. And we posited that it's a host proteins, that there's some single nucleotide polymorphism or some kind of polymorphism in the genomes of the Caucasians versus in non-Caucasians that is important in the ability of the SARS-CoV-2 to spread to East Asia or to East non-Caucasian populations. And we started our search from there. There could also be another reason, which is that because it binds to ACE2, there could be polymorphism in ACE2 gene, which is different in Caucasians. The repertoire of polymorphisms in the Caucasians could be different from the non-Caucasians. And we ruled that out very quickly because ACE2 does have certain kinds of polymorphisms, but they are very, very rare. They actually don't get up to a polymorphic state in the sense that the minor allele frequency remains at less than 0.05. So essentially monomorphic. So that we couldn't explain this pattern using ACE2 polymorphisms. So we said that let's look at TMPRSS2, and as we were, TMPRSS2 is this protein that actually cuts this cleavage site, or S1 and S2, that cleavage site is cut by TMPRSS2. So we were looking at, because because by some chance TMPRSS2 has some kind of single nucleotide polymorphisms that does not help it efficiently cut the cleavage site, then of course the gain of entry into the host cell of the virus will be retarded or will be reduced in terms of efficiency, in which case it probably will not become so dominant as as in other places. So that was just a hypothesis. We went about chasing data in order to prove or disprove the hypothesis. And in the process, what we found was that we didn't find any interesting polymorphism in TMPRSS2, but we found a polymorphism close to TMPRSS2. And in our quest to test our hypothesis, we also found that there's another gene called MX1, which does similar kinds of function, which is oriented in the opposite direction. And there are polymorphisms in the region between TMPRSS2 and MX1. And this region controls both MX2 and TMPRSS1, and there are polymorphisms there. So we went and did a thorough search of the literature, various kinds of experiments, and eventually came to the conclusion, and this is the paper that we are trying to publish, and it's come back for revision, is those are the polymorphisms that are very, very different in frequencies between Caucasians and non-Caucasians. So our hypothesis that it is the proteases, the cleaving of the S1 and S2 domains, that creates a lot of problem. And that's where the polymorphisms are, which have very different frequencies between Caucasians and non-Caucasians. So we sort of completed this entire story in the sense that we were able to figure out why the A2A is spreading so rapidly. We were able to figure out why in East Asia the spread has been somewhat different. The pattern of spread has been somewhat different compared to Caucasian populations like in Europe and United States. And we hope that this, and we were able to talk to people in the Oxford vaccine group, providing them with information that this is what's happening, that A2A is spreading, and therefore you better think about creating a vaccine that will work against A2A, because if it doesn't work against A2A, because A2A has a non-synonymous mutation, it has to work against that particular non-synonymous mutation as well, because if it doesn't, then the vaccine is going to be a failure. And they took note of our discovery, but we don't know what they have done to their vaccine, because this is big bucks. So they are not going to provide to us or any details to us. So anyway, we hope that they will take care of this. So overall then, what we have been, I hope that I've been able to impress upon you that it has an interesting evolutionary path, both in terms of its crossover from the bats to the pangolin to the human, and then how this one particular subtype of the virus dominated, and the reason for domination, and also the domination was non-uniform across the entire globe. In particular, Caucasians and non-Caucasians showed some differences in pattern, and we believe that we've been able to identify the reasons why there is a change in pattern or there's a difference in pattern, and that's what has been our contribution to this understanding, which we believe will have some impact on vaccine discovery. And also there are people who are trying to understand whether there can be a treatment for COVID-19. We also believe that our finding will help those people who are trying to find treatment, at least reduce the amount of non-response to medication against COVID-19 if it is found. So I'll stop there, and that's really what has been our contribution, and that's what my story is about evolution and spread of SARS-CoV-2. Thank you very much for your attention. I'm happy to take any questions.
Yeah, thanks a lot, Partha. So, so the attendees, you can punch in your questions in Q&A, and the panelists can, directly unmute yourself and ask your questions. So,
so Professor Partha, Himanshu here.
Hi, how you doing?
Hi. So what I wanted to ask was, there was this recent report of a Indian immigrant who went to Malaysia, and he was detected with a supposedly more virulent mutation on this COVID-19. Now, is that like, because as you mentioned, and as has been seen, the mutations are happening, but many of them may not be, so like we don't really know the virulence of many of these mutations. But so do you think uh, like, given the evolutionary pattern that this virus has shown, would it go the way SARS went? Like, it would just sort of will adapt to it, the immune system will adapt to it, or it will keep on sort of challenging the human population with new and new mutations?
So I'm no soothsayer. I won't be able to make any predictions about what's going to happen in terms of the mutations and virulence. But you brought up this Malaysian study, or this particular observation. It is exactly the same mutation that they are talking about, D614G.
Yeah.
Interestingly enough, I should tell you a little bit of history, which I really wanted to avoid. We were the first to discover the D614G and its relationship to spread. Subsequently, many of you may notice that there is a paper in Cell that's been published by the Los Alamos group and a group in the UK jointly. It's a paper published in Cell where they announced this, and that has hit headlines saying that it is their discovery. But if you look at the scheme of things or the progression of discoveries, our discovery, our publication was many months ahead of the Cell paper. This particular study, the Malaysian study, first of all, there's so much stuff coming out that nobody has the time to read.
Yeah, that is one thing. Yeah,
that's what it is. So they are talking about the same D614G and attributing it to both spread and virulence, and they provide no data on virulence.
Yeah, that
so
was the thing. Yeah,
this is a big problem that you simultaneously talk about spread and virulence and equate the two. Spread and virulence are not the same. So this is a master spreader, but is very, and we actually, for whatever little data that there are, and again, I must tell you that that there's a huge amount of sampling bias in the data, but whatever data are available, uncorrected for sampling bias, we were not able to find major differences in virulence among the 11 subtypes.
Okay.
Including this D614.
Including this one. Okay. Thank you.
There is a question from some of the panel. There is a question from panelists. Ram, you may want to ask your question.
Hi, this is Ram from THS.
Hey.
Hi, sir.
Good to see you.
Yeah. So I was just you would have noticed the Pune and the Mumbai serosurveillance data. And
Yes.
If one can kind of estimate an infection fatality rate, it may go as low as 0.1%. Whereas we have seen in Europe and in Caucasian population, the severities or the infection fatality was much higher. Do you think do you think that the genetic variations have any role to play in this? Are there any efforts?
Ram, yesterday, if you have seen the press coverage by Vinod Paul, whom you know and whom I know, Dr. Vinod Paul essentially said 1% across all of India.
Yeah.
So while it is possible that there are some communities, some regions which will have lower case fatality rate, if we average out among India where the sample sizes are large, you need large sample sizes to be able to get these reliable estimates. It's 1%. It's less than 0.1%, etc. Maybe anecdotal because of the denominator is small, so it may not really be a very robust estimate. But again, I don't know. I'm not tracking the case fatality rate at all. So it's theoretically it's possible, but I don't think we have enough evidence yet.
Yeah. Thank you.
Absolutely. There's no doubt about this. Already, you find, forget about fatalities. If you look at symptomatology, outcome of the infection, there are lots of differences, geographical differences that are already being reported. But no systematic study has been conducted trying to relate the symptom differences with the genomic differences of the hosts. So that's something that one really needs to do systematically. And being a human geneticist, my belief is that that particular level of symptomatology post-infection can be related to the genetic variation in the hosts.
Yeah, thank you, sir.
I think Manikandan has a question. Mani?
Yeah, hi. So really like to talk. So this is Manikandan, a faculty with IBSc also. So I have one question about the R0 that you asked, Dr. Partha. So
Yes.
You were saying that it's a complex statistical quantity to estimate. Do we have any estimates of the R0 of the different subtypes? Do we have enough metadata about each
So
of the
you see,
sequences?
no, we don't. We really don't, because this A2A was such a master spreader, and it spread itself so quickly. It has a huge Darwinian advantage. We haven't been able to estimate the selection coefficient, which is something that we want to do. Again, lack of time and lack of adequate data is what's preventing us. It's a huge, so in a very short period of time, after the virus goes and seeds a geographical location where you find a few of these subtypes circulating, within about 15 days or so, I mean, this master spreader has wiped out the other subtypes. So for us to get enough data to be able to estimate R0 separately for the subtypes has not been possible at all.
Okay, thanks, sir.
So, and Partha, if you look at the Q&A, there's somebody who's asked a question, which is, which type of vaccine do you think might work well? Would it be an RNA vaccine or subunit type of vaccine or whole cell vaccine and so on?
No, again, so these are questions that can only be answered by getting hold of a vaccine, administering it, and then figuring out what proportion, because everything else will just be, guesswork. So on the basis of guesswork, you design your vaccine, right? So there is this mRNA cocktail that's being done by Moderna, the US. There is the adenovirus backbone one that's being done by the UK. So these are all good guesses. India is also Bharat Biotech, and so there are some very interesting guesses that one makes. And based on these guesses, educated guesses, you go and manufacture a vaccine. Which one is going to work better? You never know ahead of time. There are too many parameters involved in terms of its efficacy that those are absolutely impossible to predict. So you should on the YouTube, Adrian Hill, who is the director of Jenner Institute, which is the Jenner Institute is actually trying to create this UK vaccine, the Oxford vaccine. He has a number of very interesting talks on the YouTube regarding what makes a vaccine tick. Can you actually predict what? And there are just too many unknowns. So the proof of the pudding will come when it's eaten, not before it's eaten.
Okay. So any other questions? So there are some other questions. So if you think something's interesting enough to quickly answer, you could in the Q&A.
Oh, is there any possibility of the virus mutating into a dangerous strain? Of course, there is a possibility, but we don't know, can't calculate that possibility or probability. Is there any relationship between pre-exposure to other viruses in the corona family? Oh, yeah. So this is very interesting that indeed, I said that 2/3 of the population must be infected, naturally infected, because we don't have a vaccine with this SARS-CoV-2. For the population to gain herd immunity and for R naught to go below 1. Many people believe, and a friend of mine who is a professor of theoretical epidemiology at the University of Oxford, she believes that, her name is Sunetra Gupta, and she's published, not yet published, but her papers are on medRxiv and bioRxiv. She's argued that we really don't need 67% of the population to be infected with this coronavirus, because many of these individuals or many of the people in the population would have been infected with flu-like symptoms, flu-like viruses, etc., and they will cross-react. So those antibodies will cross-react. So this is, she's shown some of those cross-reactions in a petri dish, so her contention is that we don't really need 67% of the population infected with the SARS coronavirus. It's going to be far less because the other people already have gained immunity because of exposure to other kinds of viruses, other kinds of lung viruses. And so the answer to your question is yes, this is most likely true. Pre-exposure to not even coronavirus. She's talking about, Sunetra is talking about of other flu-like viruses as well. Is it possible that countries having more number of individuals with comorbidities are facing more number of SARS-CoV-2 infections? As a matter of fact, this is a hypothesis that even I have. So I've started collecting some data, and I believe that your probability of recovery or death will depend on the number of comorbidities that we have. So I'm trying to collect data and do some logistic regression kind of analysis to be able to answer this exact same question that you asked. Where did it go? Does the mutations affect the diagnosis of recognition? Oh, I would not know the answer to that. What does SARS-CoV-2 have? Why does, oh, yeah, I forgot. This is a good question. I forgot to tell you that among viruses, many viruses don't have a proofreading ability after it's translated. It needs to be proofread. Otherwise, the copying errors would propagate. Many viruses don't really have the proofreading ability, but this SARS-CoV-2 has the proofreading enzymes, and therefore they are able to correct the copying errors, and that these copying errors are really translate to mutations. So that's the reason why we find the mutation rate in SARS-CoV-2 to be far less than the other similar viruses, because they have the proofreading ability.
So I think we are out of time, but there's just one last question. So this is in the chat. So there's Kirtan who's asking that there is a claim by some academics that Wuhan may not have been the original site of zoonotic transmission, but just a super spreader event. Is there enough evidence for that?
I don't know. I have no answer to this question.
Okay. And at the, and this Brinda's asking if the virus is going to mutate continuously. In that case, what would be the efficacy of the vaccine? So
yeah, so but like I said, that again, you can go to the YouTube Adrian Hill's talk, and he argues exactly what I'm going to tell you, that the mutation rate is not so high. And in any case, if the mutation rate, even if the mutation rate is high, not all of them will translate to a non-synonymous mutation, and therefore it's a non-synonymous mutation that matter in terms of vaccine efficacy. And so again, efficacy is anybody's guess.
So we'll go close with this question. Roshni asks, in India, what are the variation of the virus subtypes?
Oh, in India right now, about 3 or 4 subtypes, but again, A2A is the dominant one.
Okay, I think it's been a wonderful seminar, and I think the students and all of us learned a good deal about beyond what we've been reading on just the papers and so on, right? A scientific insight into what's really happening with COVID. So thanks a lot for for taking your time to make a presentation to us, and we'll let you get back to your the rest of the day and the symposium that you're organizing. Thank you so much, Partha. It was wonderful having you as always.
Thank you, Karthik. Thank you very much.
Thank you, Dr. Partha. See you on the Genome India meeting then.
When is that?
Whenever that is.
All right. Yes. Yes. Thank you. Thank you so much. Okay. Bye.